Now that we've talked about all the different structural elements and stages in replication of the DNA itself, we can look at this image of cell division with a little more depth. You might have a better idea and just a better understanding of just what some of these structures are. Now in the center you can see that there's some kind of finger-like structures, and if you think that those might be chromosomes, you're right. Those are chromosomes, and they're being pulled to either side of the cell. They're being pulled by these threads that aren't very visible in this picture, but there are threads pulling them from their centromeres, that center part of the cell. On the bottom right, you can see there's a cell that's very, very late in its division. You can see that the chromosomes have been torn apart, the sister chromatids are separated, and each side of the cell is now getting its share of genetic information. Now, there's also some other cells here that don't seem to be displaying that kind of clear chromosome structure that we saw when chromosomes are ready to replicate. Some of these cells have a nucleus with some kind of speckling or something's going on in them, but it's not really clear what. And that's because cells are always in a stage of development, which includes stages that aren't replication stages. There's rest stages, there's stages where the cell is growing. There's all these different stages in cell growth. And we call this the cell cycle. Now in this image you can see it looks like a big piece of pie that's cut up, but really what this is trying to illustrate is the growth cycles of cells. So let's start on the far right at G1. G1 is part of a phase called interphase. This is basically the phases that aren't mitosis and cytokinesis. So interphase is a period of growth, it's a period of chromosome duplication, but it's not a period of active cell division. G1 is a stage where the cellular contents, but not the chromosomes, are duplicated. There's also the G0 phase, which is cell cycle arrest, and that's when the cell kind of rests and it stops going through this cycle. The next phase is the S phase, and during the S phase, each of the chromosomes is duplicated. So the contents of the cell have already been duplicated. Now the genetic information within the cell is duplicated. The cell then enters the G2 phase. During the G2 phase, chromosomes are checked for errors. Cells actually possess the ability to make repairs on chromosomes, so sometimes those repairs are made. So this is the G2 phase, and these are all different phases of interphase, G1, S, and G2. So this is all the same cell undergoing these phases. You can see it's represented on the outside by the cell kind of moving along. You can see at the end of the G1 phase we have we have a pair of homologous chromosomes there, red and blue. Once the cell goes through S phase, we can see that each one of those individual homologous chromosomes is now divided, and each one has a sister chromatid. So now it looks like an X, which is really not an X. It's really two strands that are connected at the centromere. Then we continue to the G2 phase, and now our chromosomes are ready to go. They're ready to be shared and divided up. And now we enter the stage of cellular division called mitosis. Mitosis is the stage that we're going to focus a lot of our attention on. We're going to learn some vocabulary, and you'll be asked to actually be able to identify these stages when looking at a cell. But it's important to note that this is all part of this cell cycle, which is the normal growth of these somatic cells throughout the bodies of organisms. Mitosis is made up of several different stages, and I'm going to focus on 5 stages. There are other ways to divvy these stages up and include more stages, but to me, these 5 stages are a great kind of foundation to begin your understanding of mitosis. And if you want to look further into it, well, this is a great start. So those 5 stages are interphase, prophase, metaphase, anaphase, and telophase. Here in interphase, you can see that the chromatin, or like the loose genetic material, isn't condensed yet, and we still have a nuclear envelope. We also have a centrosome, and that's that dark structure on top of the nucleus here. And that centrosome is important in different stages of mitosis, but mitosis includes two centrosomes, and right now you only have one. So in interphase, if you see one centrosome, you know you're not quite in mitosis yet. You're in interphase still. Now in prophase, we can see that loose chromatin, that loose genetic information, has now been condensed into chromosomes, each one with their sister chromatid attached at the centromere. So on the left in interphase, you have these purple squiggly lines. Well, that is all organized and wrapped up, and that's what's actually making those what looks to be an X-like structure in the prophase stage. The nuclear envelope is dissolved in the prophase stage, so that's no longer there. The centrosome has also duplicated, and now we have centrosomes. And each one of those centrosomes has now moved to polar opposite ends of the cell. In metaphase, we can see that spindle fibers had begun to radiate from those centrosomes, and they're attaching to the chromosomes at their centromere. Another important feature of metaphase is that the chromosomes have lined up, and they're lined up across the kind of plane where the cell is going to divide. Each one of these chromosomes is attached to spindle fibers, and those spindle fibers are then attached to those centrosomes at polar opposite ends of the cell. During anaphase, we can see that the sister chromatids have been separated, pulled apart by those spindle fibers, and they're now moving towards polar opposite ends of the cell. And finally, in telophase, we can see that the spindle fibers go away, and we see the reformation of a new nucleus, and we can also see that the cells are starting to separate. They're starting to undergo cytokinesis, where the cells are actually pinching off and becoming two cells. Another important thing to note here is that these daughter cells are half the size of the original cell. They haven't had a chance to grow in size yet, so the actual volume has not increased. We've just divided up the information and organelles and everything within the cell into two. So now we've gone from one cell to two cells, and this process is overall called mitosis. During cytokinesis, which is a phase or a stage where the cell is actually split apart, there's a couple different pathways to splitting apart. In animal cells, we have a structure known as a cleavage furrow. And basically what happens in animal cells is that there's a ring of actin fibers, and that ring gets smaller and smaller and kind of pinches the cell towards the center, and finally that process proceeds until the two cells have been pinched apart. And we call that ring of fibers the contractile ring. It would be very similar to making a balloon animal, where you twist the balloon and then it creates two balloons. Well, in this case, instead of twisting, we have an actual ring of fibers that is getting smaller and smaller. In plant cells, Golgi vesicles coalesce at the former metaphase plate, and these vesicles fuse and form what's known as a cell plate. That cell plate is kind of like the foundation for the new cell wall, and it grows towards the outer cell walls, and when it does, it finally forms a new cell wall, and that cell wall becomes the dividing point between the two cells. So this is how prokaryotic cells divide, plant cells and animal cells. But bacteria undergo a slightly different process of cellular division, and this is called binary fission. Binary fission occurs in roughly 5 steps. One of the most important differences between the two different kinds of cells, prokaryotic and eukaryotic, is that the genetic information in a bacteria occurs in a ring. And that ring begins to replicate in 2 directions at one time, and that creates 2 rings. Another important difference between prokaryotic cells and eukaryotic cells is the presence of that nucleus, that nuclear membrane. Prokaryotic cells don't have a nuclear membrane, so once that ring of genetic information is duplicated, each one of those rings migrates to opposite ends of the cell, and then we start to see a cleavage furrow form, an area of division. And instead of a ring of actin, we now have a ring of proteins called FTSZ proteins. That ring of FTSZ proteins constricts and contracts and helps divide the bacterial cell into two new cells. These are the processes that cells use to grow and divide and to maintain the same genetic information. And so it's important for organisms to maintain the same information within the same organism. You want to keep the same kind of plan going throughout the body of an organism. You can imagine what kind of trouble you get into if your, let's say, half of your body was going by a different set of directions and plans than the other half of your body. So we call this division non-sexual cellular division, but there's another kind of cellular division that's really important in reproduction, in sexual reproduction, and also in evolution and the creation of new diversity within organisms. That process begins in another form of replication that's called meiosis. Meiosis will be the focus of our next lecture.